Hello everyone, my name is Jakub Staroń. This is next episode of MedChem Essentials, scientific seminars devoted to medicinal chemistry. Today we move to a new topic, which is fraction unbound in plasma. The focus of today's presentation will be first, plasma composition, what is plasma? Then we'll describe different plasma proteins and their properties, and finally move on to plasma protein binding. So plasma results from a centrifugation of blood. It is like the solvent of a blood. And plasma consists of 55% of the blood, and 45% are the elements which constitute to platelets, white blood cells, red blood cells. And the plasma is majority of water, but 7% constitutes of different proteins. And majority of plasma proteins, they are composed of albumins, globulins, and fibrinogen. Interestingly, these three proteins, types of proteins, they constitute to above 90% of the total weight of proteins found in the plasma. Whereas we have thousands of proteins, different types of proteins, that constitute to this 10% of the plasma proteins. But when we are talking about plasma protein binding, then our focus is on albumin and globulins. So albumin constitutes around 50% of the total proteins found in the plasma, and the following ones are alpha globulins, beta and gamma globulins. Albumin is important because it plays different roles in our body. It is being synthesized by the liver. It functions as a nutritional protein. It is rich in essential amino acids and is a transport carrier for lipids, trace elements, drugs, and hormones. The other important protein is alpha-1 acid glycoprotein, also synthesized by the liver, and it functions as a carrier for basic and neutral lipophilic compounds. So concluding, albumin transports acidic drugs, and alpha-1 acid glycoprotein transports basic and neutral drugs. ppb is expressed as a percentage of drug bound to plasma proteins. So if a drug has a ppb of 99%, means that 99% of the drugs introduced is bound to plasma proteins, and only 1% of drug is unbound. Thus, the value that we'll see in our medicinal chemistry programs is fraction unbound, which will be 1%. 1% of the drug is unbound. Let's have some illustration of that. If we introduce to the system a dose of our drug, the green circles are proteins. In high protein binding, majority of the drug will be bound to the proteins, and there will be low free drug concentration. In the low protein binding, lower quantity of our drug will be bound to the proteins, and will have higher free drug concentration. In general, more lipophilic drugs usually have higher ppb and higher hepatic clearance, whereas the renally clear drugs are hydrophilic and usually have moderate to low ppb. Fraction unbound is measured by equilibrium dialysis method in the assay of different variations. Usually the gold standard is the application of 100% plasma. However, when we are aiming into screening of a large libraries of compounds, we may want to reduce the cost and plasma requirement, and this can be achieved by reduction of the plasma content to 50 or even 10%. The measured value, fraction unbound value of different drugs in these different variations of the assay are highly comparable. The only drawback is that this reduction of plasma content renders that assay is not really suitable for highly unbound compounds, but we achieve lower cost. This is important. Fraction unbound is important because it influences measured IC50. Often this IC50 measured in the enzymatic assays is lower than in the cellular assays. I mean, we have lower in terms that the activity of our compound is higher in the enzymatic than in cellular assay. Let's say we have these two two molecules of varying fraction unbound. The first drug is highly protein bound, and the second drug has lower protein binding. So the observed IC50 in the enzyme assay will be lower. Compound will be more active than in the cellular assay. And in the second case, we will not see that much difference, because the first drug will be highly bound to the cellular components, and the free drug available to act on our biological target will be much lower. Thus, we will observe this lower activity in the cellular assay. In the experiment of the measurement of the activity of this substance, varying amounts of plasma proteins were added, 0, 2%, 10%, and 20%. And with the increasing content of plasma proteins, we see a shift of the dose-response curve, the drug becoming less active, which means we need to introduce more of the drug to get 50% inhibition or this activation of our PPR gamma. If we take into account and correction for the fraction unbound of our drug, then those all curves, they align and represent the same dose response curve as for the example with no plasma protein binding. So as you can see in the cellular assays, when we observe this shift of potency between enzymatic and cellular assays, and our compounds are less potent in the cellular assay, it may be attributed to binding to cellular components. And thus, this can be corrected if we know the fraction unbound of our drug. That's it. As a word of introduction to plasma protein binding. Thank you very much for your attention, and see you next time.